Our next speaker is Houston Wilson. He's based at the Kearney Research and Extension Center in Fresno County. He will be talking more about the seasonal ecology of Tika. So I'll turn it over to him.
Well, great. Thanks for the introduction and for the opportunity to share some of the results from our work over the past few years. I think what Frank talked about was a great setup for what I'm going to get into, so I don't need to go back over the basic biology of three-cornered alfalfa hopper. As Con mentioned, I'm an entomologist, a cooperative extension specialist in the Department of Entomology at UC Riverside, based at the Kearney Ag Center, where I work on a variety of issues in integrated pest management in orchards and vineyards. Of course, I came out of North Coast wine grape systems. That's kind of where I did my training as a grad student and postdoc, and so was pulled into all of these red blotch efforts as this issue started to kind of rear its head back in 2012, 2013. So what I'm going to present today, a couple different studies, collaborative effort with Kent Dana, a couple postdocs in his lab, Kelsey McKella, Alexis Billings, and of course farm advisor, viticulture advisor in Napa County, Monica Cooper. So three-cornered alfalfa hopper in vineyards. Should growers be concerned? Frank outlined a number of these key traits. There's a very strong association of this insect with legumes. If you look in the scientific literature, it's primarily considered a pest of peanuts, soy, and of course alfalfa, as the name would imply. They can feed in oviposit on grapes, but cannot complete their life cycle on grapes. So kind of based on just that stuff alone, the conclusion is that they are considered a relatively minor pest of grapes, and you see this in the grape production manuals that the UC puts out. That said, we see them in vineyards. I've seen this insect the entire time that I've been doing vineyard entomology, but I've always been focused on other insects like grape leafhoppers, mealybugs, things like that. So all that said, of course, Tika can transmit red blotch virus under a greenhouse setting, and that's a really key component. And again, Frank was kind of alluding to some of that, thinking about the transmission efficiency, the acquisition time, things like that. But we do know that it can move this virus between vines, and there's a series of studies that have associated the incidence and spread of red blotch virus with this insect. So what I'm going to talk about today really are two key research questions that stemmed out of some of the initial early observations of this insect, especially once we knew that it could potentially move the virus. So the first question, does Tika aggregate at the vineyard edge? A couple studies gave some evidence for that. And then second question, can ground cover management influence Tika activity in the vine canopy? So we'll start off with this first question. There is evidence from previous studies showing some spatial clustering at times at certain sites, typically adjacent to riparian habitat, and then in some cases that clustering is associated with increased incidence of red blotch virus. Why would that be? Given their ecology, we do know that they overwinter as an adult and potentially in sheltered habitat in a riparian area, or maybe there's alternative leguminous host plants that are available to them there. This image from a paper out of Mark Fuchs' lab, showing two different sort of spatial aggregations of red blotch virus in very proximal vineyards, of course. So it's not always the case, but we do sometimes see this. And so it was a narrative that was kind of developing around the science of this insect. And so we set out to conduct a study to more closely evaluate that. So it's a 2-year study, all in vineyards with red varieties. These are well-established vines, big flat square blocks with uniform irrigation, pesticide-free, all the kind of basic stuff you want for an insect study in vineyards. There were a total of 5 sites, all of them adjacent to riparian habitat throughout Napa County. And what we did was sample along transects, taking samples at the edge of the vineyard, about 10 meters away from the riparian habitat, so just across the road, basically. You usually have a little dirt road over there. And then at the interior of the vineyard, about 150 meters or 150 yards out into the vineyard. At each of those edge and interior sample sites, we put up yellow sticky traps in the vine canopy. We replace these every 2 weeks approximately, between March and November. We also started looking for petiole girdling 1 or 2 times a month from June to October. And then at the same time, we also recorded the percentage of ground cover in the areas at the edge versus the interior, and conducted a qualitative ranking on a scale of 1 to 4 of how desiccated effectively that ground cover was. So it gets a ranking of 1, it's big healthy green stand. If it gets a ranking of 4, it's mostly dead. So you'll see where that comes in here in a minute. Focal organisms for this study, of course, were three-cornered alfalfa hopper. We also focused on western grape leaf hopper sort of as a control insect that we understand its ecology, especially in relation to these natural habitats. And then we also were monitoring for the sharp-nosed leafhopper, Scaphitopius corneticus. Frank alluded to this in some of his work and had sampled it, and others are looking at different species within this genus. There's work we've been conducting. I'll show a few of the results at the very end here, but essentially indicating, giving us some good signs that the Scaphitopius is a potential candidate vector. It's worthy of further investigation, so we included it in all of these studies that I'm going to talk about here today. So, setup, 2-year study, 5 sites, all this hard work, and one graph basically for each insect, but a lot of information here. So, three-cornered alfalfa hopper, no clear edge effect whatsoever. The populations overall seem to be sort of split between the ground cover and the vine canopy. So what you're seeing here are the mean organisms per trap per week in the early versus the late season. This is just when we see the 2 population peaks of this insect. And then in the yellow, that's at the edge, and then blue is at the interior, and then you have the hatch. Ooh, this pointer's a little funky. The hatched bars it's the populations on the ground cover, and the solid ones that's in the canopy. So not a very clear edge effect for Tika here. With grape leaf hopper, these are trends that we would basically expect. You can see early in the season they tend to be more closely associated with ground covers. Later in the season, more closely associated with the canopy, and then you do see increased populations at the interior of the vineyard compared to the edge. So western grape leaf hopper overwinters in a reproductive diapause down in leaf litter on the vineyard floor. As the grapevines start to push out leaves, they become active and move up into the canopy. The way we were sampling with sticky cards, that resulted in increased capture of those leaf hoppers down in that kind of ground cover area early in the season as they made their way up to the vine. And then you see here later in the season much more activity on the vine itself. This is considered a key pest of grapes in California. It's a native insect to the state, closely associated with grapevines, so all of that makes sense to us. The increased populations at the interior relative to the edge, there does seem to be somewhat of an edge effect here, and this actually was something that bore out in an earlier study conducted by our lab looking at populations of grape leafhopper and their parasitoids, these little anagrous parasitoids that attack their eggs in vineyards adjacent to riparian habitat. And what came out in those earlier studies, and what we think would explain the trends we're seeing here, is differences in grapevine vigor at the edge versus the interior of the vineyard. So as you move further away from that riparian edge, there, as in our previous studies, has tended to be increased vine vigor, increased free nitrogen, and grape leafhoppers respond very much to that. So they're looking for more vigorous vines. So that kind of all made sense to us here. And then finally, the last insect, Scaphitopius, no edge effect, but a very persistent population in the canopy. So unlike the other cicadella, the grape leafhopper, we don't see early season activity really strongly tied to the ground covers. We don't really know where this insect overwinters. Very little is known about this species. The only records that exist are from grapevines. I'll talk a little bit more about that later, but what you can see here is both a lack of an edge effect and a very clear association with the grapevine canopy, both early in the season and later in the season. Again, some of this was sort of mentioned in Frank's talk, this relationship between Tika activity in the vine canopy and petiole girdling. We know that's damage that is caused by three-cornered alfalfa hopper. And what this is showing is the population of Tika in the canopy at the edge, in the interior, those solid lines. And then you can see the appearance of petiole girdling in both of those areas shortly thereafter. So as they move into the canopy, you start to see increased activity, they start feeding, and then those girdles appear shortly thereafter. This is our year 1 data. Here's our year 2 data. Similar trend, much more pronounced in the second year, so interannual variation in their populations, but definitely a thing. So move into the canopy, they start feeding, it's girdling, that's when you'd have potential for virus transmission. When we look at the relationship of the ground cover status to the activity of these insects in the vine canopy, we do see a fairly strong relationship for all of them, but quite strong for Tika. As the proportion of ground covers that are mostly dead increases, the overall proportion of the population of that insect in the vine canopy also increases. And from our observations and from just reviewing the literature again, this insect is very closely tied to legumes. It can't complete its life cycle on grapevines, and so in my experience, when we're out in the field sampling and looking at plots with ground covers that are slowly desiccating or they're being mowed or something like that, this insect loves legumes. It doesn't want to let them go. When we mow them, when they dry down, you have a big, healthy grapevine canopy all full of, plant material and water and all that stuff, and little Tika are down there on this scraggly little legume holding on for dear life. So it's really a forced movement, we think, up into the canopy. Similar, well, not as exacerbated, but a positive trend for these other insects as well. So summary of key findings from this study: three-cornered alfalfa hopper, they don't appear to aggregate to the vineyard edge. They hang around on ground covers as long as they can. Movement into the canopy is followed by girdling. This has been shown elsewhere. Overall, very low populations. Out of these 3 insects, this was the least frequently encountered. Western grapeleaf hopper, a lot is known about this insect. They overwinter in the leaf litter. They're active around the ground covers in the spring. Their reproduction is in the vine canopy, and they are key pests of grapes. There's very high populations at the interior. Again, this has been borne out in other studies as well. Scaphatopius, no edge effect, least active of all 3 of these on ground covers, consistently associated with the vine canopy, and as I mentioned earlier, grape is the only known host for this insect. Okay, so question number 1, knock that out of the way. Question number 2, can ground cover management influence Tika activity? So Frank gave a nice outline of this already. The Tika overwinter as adults. They feed and oviposit on ground covers in the spring, primarily legumes. First generation nymphs develop on those ground covers, these little furry, spiky guys here. They complete development on those ground covers just as they start to dry out, typically, at least here in the North Coast in the conditions we have. And as they molt into an adult, those adults are the ones that move up into the canopy. And it's important to note that while the adults are highly mobile, the nymphs are not. They can't fly, they don't have wings. I suppose they could walk down off their ground cover and walk up into the vine, but they're not as jumpy, I guess you could say, as the adults. And that's an important thing to consider, because when they're down in that nymphal stage, developing on those ground covers, you may have an opportunity to knock them out. And so that was kind of the impetus for this study. Could disking ground covers reduce canopy activity by eliminating the nymphs? So another 2-year study, similar type of setup, red varieties, established vines, flat level ground, no pesticide. We had 5 sites in year 1, 3 sites in year 2. The ground covers were a blend of legumes and grasses. These are intentionally sown cover crops with some weedy vegetation mixed in where establishment was spotty. And what we did at each site was pair together sets of 5 rows and have 5 replicates of that per site, and then randomly assign a plot to be mown versus mown in discs. So mowing in the spring versus mowing and entirely eliminating that vegetation from those plots. Insect sampling, a lot of the same things as what we saw in the previous study. Yellow sticky traps in the vine canopy, check those every 2 weeks, and we focused on Tika, grape leafhopper, and Scaphatopius. So again, we'll go insect by insect here. What we saw with 3-cornered alfalfa hopper was that disking does tend to reduce their activity in the vine canopy. Not always, but more frequently than the other insects we encountered. So that does give us some indication that getting rid of that vegetation entirely has a further downward pressure, puts a further downward pressure on their populations and their ability to ultimately kind of send adults up into the canopy. Western grape leafhopper, we saw that disking actually promotes more activity in the canopy. And again, this sort of goes back to that vigor hypothesis, and we've seen in previous studies looking at cover crops, which were intended to increase natural enemies, beneficial insects, to then go predate or parasitize grape leafhoppers. And in some of those studies, we've seen that there were lower overall populations of leafhoppers in plots that had cover crops, not because of a natural enemy effect, but because it was devigoring the vines. And we know that these leafhoppers really like more vigorous vines. And so in this study, getting entirely rid of those ground covers versus just mowing may have potentially created a similar effect. We didn't measure vine vigor or petiole nitrogen or anything like that, but just, you know, as somebody who's worked on this insect and with ground covers for this insect for many years, that's kind of how I'm interpreting that data. Finally, Scaphitopius, not affected by ground cover management whatsoever. And again, this just reinforces the close association that this insect has with the grapevine. They're not moving to ground covers. They're not, as those things desiccate, moving back into the canopy. They start off in the canopy and seem to stay there all season. And if we mow and if we disc, we don't do anything at all, it doesn't really seem to influence their population. So again, just driving home this point, they're very closely associated with the vine canopy. So summarizing all that, total elimination of ground covers reduces teak activity in the canopy. The timing, of course, could be improved to more closely track the nymphs. And I think Cindy's going to talk a little bit about this later. What we did in this study, growers were going in at the normal spring timing that worked for them to deal with their ground covers, either mow or disc. If you were to more closely monitor for the presence of nymphs and kind of get all of those eggs that have been laid for that first generation to hatch out and get as many nymphs as you can on those covers and then say, okay, now's the time, let's go hit them, I bet you would probably get a more pronounced effect. In this case, again, we just did it at the normal timing that worked for the growers, which approximately coincided when you would have nymphs out there. Grape leafhopper, close association with the vine canopy, possible vigor effect with disking, and then Scaphitopius very closely associated with grapevine and unaffected by ground cover management. So these two questions coming back, do teak aggregate at the vineyard edge? No. Can ground cover management influence teak activity in the canopy? Yes. So we summarized all this in a couple of recent publications, just showing those here. Be happy to share those with anybody. Just come talk to me afterwards. And then concluding remarks on three-cornered alfalfa hoppers. So they can transmit red blotch in a greenhouse, but transmission efficiency and field transmission do remain unclear. Again, Frank did a nice job talking about some of the nuances of that. They do have this fundamental relationship to ground covers and legumes in particular. They can't fully develop on grapevines, so it's a very difficult situation in terms of blaming them for potentially all of the insect vectoring that we're possibly seeing of this virus. All that said, they're still a concern for growers. I understand that, and rather than eliminate legumes entirely from overwintering cover crop blends, which was an idea put forward very early on when we first started to think about this insect, it does seem that the timing of mowing and disking could help to manage them. I'm pointing out a table here from a recent paper that we published that's a survey of both insects and plants to identify potential vectors and host plant repositories of red blotch. And what I'm highlighting here is there's Tika down there at the bottom. I'm sorry, it's pretty small on the screen, I realize. And then here's Scaphitopius. What I'm trying to highlight here is that overall about 15% of the Tika we captured tested positive for red blotch. And this wasn't salivary glands, this was a whole body analysis, so you can take that at value that as you may. And then Scaphitopius, 58% of them tested positive. So again, just adding to this evidence that we think this would be an insect that's worthwhile of closer investigation. Little is known about Scaphitopius. There's scant records. The few that exist are from Grape, citing a study here that talked about this insect many decades ago. Our studies show a high affinity for grapevines, and they do frequently test positive. That's shown in this table here. And transmission studies are underway, so I'm kind of setting up a little segue into what Kent is going to talk about here next. So with that, just want to say thanks again for the invitation to share some of this information with you all. I want to acknowledge our funders, the CDFA PD GWIS Board and American Vineyard Foundation, a whole host of people from UC Cooperative Extension, UC Berkeley, UC Davis, and then of course, as always, numerous growers and PCAs throughout the North Coast. So got my contact info there, and thanks again for your time.
Actually, we're early. We have time for questions if you have any questions for Houston. Any questions from live stream online? Nope. He's here.
I have a quick question.
Oh.
Is Scaphotopius associated with damage to the grapevine?
Yeah, so Kent's lab has been holding them in colony and making observations on the damage that they're causing to the vines there, and it seems to be similar to the leaf stippling that we see from grape leafhoppers. I think Kent can probably give more details on that. This species, I've seen it because of that big sharp nose that's always stood out from the first year I did research on insects in vineyards. We've never seen, you rarely encounter the nymphs, and now that we're holding them, we know they jump away, and so it's something you don't quite commonly encounter even though they're there. And I think in many cases, as I'm understanding the sort of damage they cause now, thinking back on experiences where, you know, I've been monitoring grape leafhoppers turning over leaves in vineyards for almost a decade now, there may have been stippling in some cases that was from Scaphotopius, but just sort of defaulted to grape leafhopper. So it's something that I would like us to start paying a little more closer attention to out in our scouting. But you know, if there's nymphs on there from Scaphotopius making that sort of damage, if you pick up a leaf, you're probably not going to see them because they're just going to kind of flick away really quick. Probably more of an answer than you wanted, but yeah, they cause some damage. Okay, well, I'll be around for the rest of the day if y'all want to talk. Thanks.